All right. Hello, everyone. Welcome to our webinar on modular analysis. Very excited to present today. Let me go ahead and share my screen so we can go ahead and get started. So I'm really excited to present this. This has been something that we've been working on with our clients for months now. Modular analysis is a new capability in the Ganymede platform to build out and offer very highly specialized analysis tools directly linked to your instruments and apps. This is something that has been a high demand thing and is already yielding some pretty impressive results with our client base. So as a round of introductions quickly, we have on the line Nick Flores, one of our scientific software engineers, Olivia Watson from our manufacturing software engineering team, and Andrew Mecca, one of our scientific implementation managers. I'm Nathan. I'm one of our co-founders, and on the Ganymede side, manage our commercial and solutions teams. So let's talk a little bit about what modular analysis is before we get into the actual demo. As a quick refresher on the Ganymede platform itself, we do three major things. We connect directly to your lab instruments or other data sources in your lab. We, on lab instruments, will install agents on Windows PCs or connect with OPC data sources wherever the data is. With software applications like analysis tools or ELN and LIMS systems, we can connect with APIs, we can work with files, we can connect with databases, you name it. Our job is to get the data out of the instruments into the cloud. We will parse it, standardize it, and then historically, a lot of what we've done from there is we analyze the data and then can automate pushing it into something like an ELN or LIMS system purely in the back end in an automated way with our pipelines, our flows. And that's a very powerful approach when things can be standardized. But what we found is that so many aspects of science can't be standardized. So much of the work is a human discretionary thing because there is in scientists' heads real unique context about the experiment, about what's going on, about the study. And so there is a lot of work that needs to be done in these human interactive analysis tools or in Excel. And so our approach over the years has increasingly evolved more and more towards getting people into the loop with these workflows. And what we've done now with modular analysis is realize the final form of that, where we can now build these fully interactive analysis applications directly in the Ganymede platform, linked directly to your lab instruments and linked directly to your ELN and LIMS systems to maximize context and make this as smooth an experience as possible. Two things I want to note about this that make this a unique approach. One is that this is a highly customizable approach. And so we can offer out of the box, not just a generic curve fitting app for things like IC50 or EC50 dose response curves, but something specific to ELISA or something specific to like a reporter gene assay that gets you much more context and much more scientific insight to your workflow. And also smooths the data entry because we know much more context about what particular type of thing this is. This is, I would say, something that we want to be a very tightly coupled thing to the workflow for high scale workflows or workflows where you do know what the assay is enough to start standardizing it and automating it. We don't necessarily want this to try to supplant dedicated tools like a GraphPad or like a FlowJo for flow cytometry. Those tools are great and have been built out extensively over the years to handle a lot of edge cases and be great for exploring data. But once you are starting to get tired of doing analyses repetitively, or you have some particular analysis use case that's more unique, that's where we can come in. The other side of it is we have constantly had clients come to us and say, there's so much hype out there about AI, and we have so much data that we're capturing on the scientific side, especially if they're already working with Ganymede. Now they have so much data in the cloud. How can we leverage AI in a practical way in the lab and actually realize it? And this to us is the path. It's not having it be something that runs silently in the back end as a magic wand, as much as something that is actually at users' fingertips in the application, being in this human in the loop step of being in the app where we can partially fill out analysis for humans with AI or even fully automate it, makes it much more of a conversation with the data, which is what's needed to really leverage AI. And so we found that in cases like chromatography, for instance, we've been able to partially or fully automate peak finding to recommend peaks to our end users rather than them having to repetitively fill them out every time. Or even change it to a review by exception workflow where the peaks are just there and they are saying yes, or there's exceptions and they say, oh, this looks like something I want to investigate more. It's really transformative in terms of being able to actually leverage that automation. So with that, I'll turn it over to Andrew. Andrew, I think you're on as well right now, right?
Yep.
All
All
right.
right. Thanks, Nathan. Nathan just gave kind of an overview of the Ganymede platform as a whole and some of the capabilities here. I'm going to spend a couple slides talking specifically about what is Ganymede's solution for bioprocess data. And through the demos today, what we'll see is our modular analysis application for upstream bioprocess, which Nick Flores is going to present in just a little bit. But just to kind of walk us through what our solution is for bioprocess. With the Ganymede platform and our modular analysis app, really what we can do here is end-to-end automation. And this is all the way from raw data files coming off of a bioreactor or a cell culture analyzer, all the way to fully analyzed, visualized data that is ready to be exported and put into a batch record, as an example. So what our solution is it encompasses that from A to Z. So we start with the data collection, which I think Nathan already briefly touched on. But essentially, we'll be pulling off raw data from all connected instruments that are involved in your upstream bioprocessing run. So, and I just have two example instruments here, bioreactor and cell culture analyzer. Next, what will happen is all of this raw data will be cleaned up and parsed into structured data tables in Ganymede. And then these data tables will feed into our modular analysis application, which has out-of-the-box automated analyses and visualization tools to view all of your aggregated data together in one place. Next slide, please. So the demo context that Nick is going to take us through is the following. So I can imagine that I'm an upstream process scientist working to optimize the yield of a scale-up step for a new drug candidate that I'm working on. What would be great for me as a scientist is if I could be able to quickly identify the critical process parameters for a given experiment that have the greatest impact on product yield. And I'll use this information to inform how I'm going to design my next set of experiments. And so how Ganymede can help with this through our end-to-end automation here is faster decision making. And so in the demo, we're going to show 4 key components. Automated data pickup and parsing from really any data source, online, at-line instruments. Perform automated analysis of key critical process parameters from that data. Perform validation and cleanup, such as outlier detection and others, and then visualize that data in real time.
All right. Cool. Thank you, Andrew. And I will turn it over to Nick to go through the platform.
Awesome. Are you seeing my screen?
Yes.
Looks good.
Great. Okay. So thanks, Andrew and Nathan. So like Andrew said, I'm going to try to give you a walkthrough of the Ganymede platform and really some more concrete detail and basis to think about how Ganymede can help in automating your bioprocess analyses. What we're looking at right now is the Ganymede homepage, which is effectively the hub for you to interact with any facet of the Ganymede platform you're interested in. What we're going to go to first is our connections page, where we're able to see live instances of Ganymede agents installed on lab PCs. An agent, just to frame your thinking, really can be thought of as an executable that's on a lab PC listening for the generation of new data. When it finds a new data file or a new line of data, it contextualizes that information with tags, which we'll see, and uploads it to Ganymede for long-term storage. So we're seeing 2 examples of agents that we've designed and configured here, one for a Vanquish HPLC and one for an Amber 250. If we were to investigate, for example, the Amber 250 agent connection in detail, we'll see that we're applying specific tags like the Department of Upstream Fermentation or the lab space of Lab 1027 to all data that comes off of this Amber 250 system. Now, beyond just globally applying tags to a specific agent or connection, what we're able to do is parse, in a lot of cases, the actual files or data coming off itself to extract things like your method name and contextualize that data again in our ultimate kind of data lake. Now, the way files look in Ganymede, and like in this file browser, which we'll see here as it's loading, where you see a number of files that have been uploaded by agents, contextualized by the tags that are located here and I'm hovering over. This gives you a clean interface to filter your data and kind of download and explore older data sets and understand exactly when they were uploaded and how they were generated. Now, agents are there at the forefront, right? Gathering your data and contextualizing it But more than just upload and store our data, we want to be able to analyze it. And for that, we're going to leverage what are called Ganymede flows. So I'm in the flow editor window right now, and I'm going to open our bioprocess data analysis and modeling flow. I'm going to zoom in a little bit to give you a slightly better view. And so what flows can really be thought of as is really individual nodes that are running just Python code to analyze your data. If I were to just quickly inspect an individual node, I'll see the actual code that is running when this node is hit and activated. Really, the reason I show you this is to understand what Nathan said, which is that these flows and analyses are highly customizable and tailored to your specific needs. So while I'm going to go through specific steps that we've set up for our bioprocess demo today, understand that your process may look a little different and these flows can be configured to meet your needs. Now, looking specifically at this bioprocess flow, what we see first is that we're ingesting data from two different sources. Our AMBER 250 data that we collected, which is going to have our online parameters, as well as our data from our HPLC Vanquish, which is going to be measuring things like our metabolites. Together, that data is then going to go further through our flow to activate things like our anomaly detection node, do growth curve modeling, some correlation analyses, and ultimately a partially squared analysis to try to predict the outcome of these fermentation experiments. And it's going to end in uploading your data to your ELN. While the flows, like I said, are highly configurable, they're almost always going to end in this upload to ELN step, right? To make sure that we maintain data traceability throughout the start of that raw data generation to its final analyzed state in your ELN. Now, since we're talking about bioprocess, I do want to hone in on one specific feature of these flows, which is really sourcing from our anomaly detection. What I want to highlight is that, like Andrew said, we're able to do real-time analysis of our data as it's coming off of your bioreactor. And so what we've set up is a statistical process control methodology to understand when a particular parameter is outside of spec. What that can do is send an email notification, which I'll hop over to an email real quick. Please don't judge the state of my inbox. But if we see this email here, this was triggered by this flow this morning at 10:12 a.m. when I triggered it. And it's detailing what flow sent this alert, what parameters exceeded our thresholds and what those thresholds were, and the Ganymede context for us to further investigate in Ganymede and outside to understand if we need to take corrective action in the real world or if things are okay and we can just continue to monitor closely. Okay, so that was a very fast, I know, brief explanation of agents and flows. And really these two kind of concepts together are how we automate our data analysis. But like Nathan said, there's often times when you need to leverage specific scientific expertise to modify an analysis, or like you said, maybe to just approve it to say, yes, this looks good. Additionally, in the context of bioprocess data, you're often trying to pair varied sources and visualize them in a single dashboard so that you can fully understand the context of your experiment. And so it's with these two motivating factors that we've designed the upstream bioprocess modular analysis app, which I'll go ahead and swap to right now. What we're looking at here, again, is a visualization of all of our data that is being populated via Ganymede flows. So please understand that the underlying source for all of these charts that we're going to see is the outcome of those flows. Now, what we're looking at first here is our online data. So we can see things like our temperature and pH and how they varied not only in individual runs, but overlay many runs to try to really discern if there are trends globally. What we also see is a direct visualization of that anomaly detection step, where we're saying what were our thresholds, what points triggered them, and what might have triggered our email alerts, and maybe how to reconfigure them if we aren't happy with the current behavior. Additionally, we have visualizations, like we said, for the at-line data for things that are coming out, for example, your HPLC or potentially a growth analyzer, really allowing you to freely explore your time series data in a way that makes sense to you. The final tab here is just exploring your product titer data, which might be the outcome of your fermentation if you're just trying to produce the most quantity of a particular product. Beyond this sort of typical dashboarding that is, I think, fundamental to your bioprocess analysis, what we also offer is a little more detail, again, and an opportunity for scientists to take action on your actual analyses. What we're looking at here specifically is your cell growth modeling, which you're going to be using to calculate things like your specific growth rate. And you're seeing what the flow thought was the best model to use and the approximate linear range. Well, let's say as a scientist, I believe that a different model fits better, and I believe that the actual linear growth range is somewhere around here. I can freely make those edits and submit those changes back to my ELN. Now, the key thing to note, right, is that we want to maintain traceability. And so this submit changes is not just saying I committed changes to my data, it's saying when did I do it? Who made these changes? So that you can really understand your data from the automated standpoint and whatever human interventions were made. Now, beyond basic cell culture growth kinetics, we also have, like Andrew said, an investigation tab for critical process parameters. So cases where you might want to investigate the correlation between key set points in your bioreactor and your key outputs, and you can explore them on an experiment by experiment basis. Additionally, as you generate significant quantities of experiments, you start being able to do things like PCA analysis, where you can look for groupings among experiments to say, oh, there's a cohort that's interesting here. Maybe I should pull up those overlays together and analyze them in my time series dashboard. Now, while I will say this tab is very focused on sort of hypotheses generation and data exploration, a common concrete ask that we see with bioprocess experiments is folks want the ability to model their experiments based off of historical runs. And so what we're seeing here is the direct output of our partial least squared analysis node and the model that it generated based off of analyzing all of our historical data. You're able to see things like how well our current model is performing, explore the contours of particular variables against other ones and how that affects your key output metric. You additionally see details on the model, which is going to highlight variables that you might want to focus on or change in your further experiments to try to drive the growth you desire in your bioprocess. Now, again, this slide here is focused on review, but really you want to be able to take action based off of this model that we've built. And so with that in mind, we designed our experimental design tab, our final tab of the bioprocess app we're going to explore, to take that model and give you an intuitive visualization of what we expect your experiment to look like if you set your parameters at certain set points. What it's doing is it's saying, based off of that model, I can get a rough range that we think is intelligent to explore based off of what you want to optimize and give you our expectation for the range of outcomes. Just like our growth analysis, our model may not be perfect, and so you may have intuition to say, oh, that temperature set point is too high, I need to adjust that, and you can see in real time how that affects your predicted outcomes. Now, this is great for exploring your model, but ultimately you want to use it, like I said, to design your next experiment, which you can only do by saying, I need to specify a particular sampling strategy for how I'm going to construct kind of my individual reactors, and I need to make sure that that is designed in with the specific reactor model in mind. Am I going to have 12 reactors or am I going to have 24? That's going to directly affect how you're able to explore this sample space. And so after making these changes and being happy with your experimental setup, you're able to, again, use the submit button to submit your detailed experimental design to your ELN for you to further feed this system and this ever-present design build test loop of you trying to optimize your bioprocess and build a model that's better and better and more accurate. And so with that, I will turn it back over to Nathan to go through the rest of the
All right.
seminar.
Cool, thank you. So let's just quickly recap here what we just saw. Let me go ahead and share my screen again. So hopefully that gives you a good overview of the modular analysis system. We're taking the same data that we connect and standardize through Ganymede, but now being able to knit it together in a much more built-out app experience that has a lot more capabilities to both apply modeling and have a user interactive component. You saw data go from the automated file pickup, or with bioreactors, we can also work with things like OPC and contextualization through parsing, and then data validation, and then finally into this visualization layer. There's a question I saw briefly that I'll speak to around how whether the vendor customizes this or if customers are enabled to do this themselves. The short answer is that customers can do this themselves if they like. Our modular analysis apps are pretty out of the box for a lot of use cases, and our goal is to make them as out of the box as they can be. And then we as the vendor can customize them if you want. But the goal is to make it so that customers can also customize this, because if you say, I have this particular type of cell culture that's different and I need to change the models that are available, or I need some new chart, that's the modular part of modular analysis, is that everything that you see is defined in pretty straightforward low code in the Ganymede lab as code system. And so you can go ahead and go in and switch out these components at a component level and change that app experience almost overnight. So it's a very effective way to make these apps and analyses work for your science, which changes naturally. But yeah, we, like everything in Ganymede, we offer a lot out of the box, and we'll support the customization as needed so that you don't need to worry about it. But we always do say that customers are more than welcome to build on it themselves, especially if you have engineers for that. Up next, we will actually give a preview of the chromatography application that Olivia and Nick have built out. We're also super excited about this because this is much more of a user interactive workflow and should give you a sense of both how the user interactivity will work in these systems, and also a little bit more of where AI can come in to actually pre-populate some of the assay parameters or analysis configurations. So to give a brief overview of our chromatography capabilities, we'll connect directly to chromatography systems, or can parse data out of chromatography data systems and connect to that, build a standardized data layer out of that. And then this is where, with that data, we can either partially or fully predict things like peaks or things like the structure of methods, and either directly automate that entirely, or more often put this into a system like modular analysis that can then present this to the user and say, here are the peaks, like, does this look good? Do you want to modify them, etc.? And then with that, we can either write that data out, the peaks and parameters, or whatever you're looking for in that analysis, into an ELN or LIMS system. And in turn, actually, we can integrate data out of ELN and LIMS systems back into an instrument and create a work list. Or in some cases, we can actually write it back to the chromatography data system directly. So if you really like something like Empower or Chameleon that you want to store the data, including the analyzed peaks in and the analysis, we can write back things like methods into those systems as well. So true bidirectional capability here, with as much machine learning and AI as you want, depending on how much can be predicted about your analysis. So this is a great, genuine, practical application of AI that we found in the wet lab. But I will turn it over to Olivia now. If Olivia, you want to go through the chromatography system.
Sounds good. So I'm going to jump straight into our modular analysis application for chromatography. But similar to the way the bioprocess demo was that Nick just walked through, we have an agent running in the background that's monitoring a local instrument directory for any new files. And once it sees that, it's uploading to Ganymede and then kicking off our chromatography flow. What that flow is doing in this case is parsing that raw chromatogram data and writing that to a table in Ganymede. And then we have a peak finding algorithm that's applied to this raw chromatogram that's finding our peaks. And it's also applying calibration curves and then writing all of that data back to Ganymede tables at the end of the day. So you can see here we have our raw chromatogram data and the peaks that it found. We have an instrument drop down, so you can select which experiment you want to look at, and then which sample from that experiment to display. You can see here that we did modify the peaks that were found to be a little bit rough around the edges so that we can kind of demonstrate the different modifications and adjustments that you can make. But these were the peaks that were found with our automated analysis. You can see they're like highlighted and identified here. And then we have a table at the bottom that has the peak area and height and things that were calculated by our algorithm. We've also identified two of these main peaks, glucose and sucrose. So that's as each sample is run, we're identifying, we know to expect glucose in this range. And so we're kind of identifying that. So one of the first adjustments you can make is if the algorithm misidentifies a peak, you can come down here and change the name of whatever peak was found to whatever you know it to be. Say if it's in a different range than expected and it's misidentified, you can correct that identification. We have a handy button here for resetting the data. What this button does is it resets the data to what was initially found by the analysis. So any changes that were made, if you made them in mistake, you can reset to what it was initially. You can see up here we have a list of kind of the actions that you can take to make these adjustments as necessary. Hopefully the algorithm works as expected and as desired to identify these peaks, but for the cases of outliers or just if something went wrong in the process and the algorithm misidentified, you can come in and have a human in the loop come make these adjustments. This first one here is editing the baseline. You can see in this data, we have a fairly linear baseline right around the 0 mark. But say if you have a high signal to noise ratio and you don't want to include that noise in your peak areas, or if you have a drifting baseline that wasn't correctly accounted for in the algorithm, just something where you need to come modify that baseline, you can just simply draw a new line and click commit. You can see it readjusted that peak area automatically, and it also adjusted this calculated peak area down here at the bottom. So you can kind of do this on a peak by peak basis if one of your peaks just has a weird drift line that you need to account for, or you can do it for the entire chromatogram at once. And again, you can reset back to what the initial baseline was. This next action we can take is splitting a peak. So you can see this third peak off to the right probably should be split into two. So maybe the two peaks are too close together for the algorithm to identify them separately. Maybe one of them was too small for it to be picked up individually or anything like that. So you can come in and split those out into two separate peaks. So what you can do is just select where you expect that split to be. And you can split them by a simple drop line, or you can also split them by a tangent skim. And so this could be the case if you have like a main peak that just has shoulder peaks off to the side. You want to skim off that shoulder peak, but you want the majority of the area under that peak to account towards the main peak's peak area. So you can use the tangential skim to just skim off that shoulder peak. You can see it adds the new peak down to this table at the bottom and automatically calculates those values. The next action we have is updating boundaries. So you can see on this first peak, we probably want these start and end times of the peak to extend down to where the peak meets the baseline. So what you can do here is simply select that peak. You can see it kind of highlights it, and then you can select your new desired start and end times and click commit. It automatically updates the area in the plot as well as in the table down here. And again, you can reset it to what was initially identified. Our next action is adding a peak. So say if there's a small peak or that was smaller than expected, or maybe it was in a relatively high signal to noise ratio area, or just something like that where the algorithm didn't identify it as a peak, but you with your scientist intuition know that it should be identified and integrated. What you can do is come in and select where you expect that peak to be. You can click add. It'll automatically integrate that area, find a start and end time for it, and then add it to the table down here as well. And then on the opposite side, if we found a peak with the algorithm that we don't expect to be integrated and don't want integrated, we also have a remove peak function. So that could be if there's a really high signal to noise ratio and maybe the algorithm picked up some of that noise as a peak, or there's a buffer peak that maybe had a later retention time than expected, and so it was picked up when it shouldn't have been. What you can do here is come and select that peak. Again, it's highlighted, and you can click remove, and it'll remove it from the table and remove that area from any of the area percentage calculations that you may make. So that's kind of an overview of all the different adjustments you can make if any need to be made after the algorithm runs. We do have down here this publish data button. And so once you're satisfied with these adjustments you've made, you can click that publish data button. And like Nick said, for the bioprocessing app, this is going to record all of these adjustments that were made, the new peak areas that were found, who made them and when, and then publish to your ELN or system of record. So this is kind of how you can interact with this data on a sample by sample basis. And then you can see up here at the top, we also have a couple kind of higher level overview tabs where you can look at the data as a whole. This first one for the regression analysis, these are the calibration curves that we're applying to the samples that are run. So you can see we have known standards at certain concentrations for both of the molecules that we identified. So what you can do here is you can see glucose is a pretty good fit with the power model here. You can see it fits the data pretty well. And so any new samples that come in where we identify a glucose peak, we will compare it to this calibration curve to calculate that concentration. But if we go to sucrose, you can see that this power model isn't quite a good fit. Yeah, it's not fitting the data as expected or as wanted. So what you can do is come to this model select and actually select which different model you want to use for this data. And similarly to Bioprocess, once you click publish, it'll record this model in Ganymede, and then any future samples that come in with these molecules identified will be compared against the new model that was selected. So and like we said for Bioprocess, you can come in and add different models as needed or anything. But yeah, you can see that the linear is probably a better fit for this molecule, and that's probably what you want to use to identify any new samples coming in. This next tab is our experiment summary tab. You can see you can kind of select the different experiments from this dropdown and then the different molecules that were identified. On this tab, we have a few control charts. You can see we have one for retention time, peak width, and peak symmetry. So what we've done here is we've identified ranges that we expect these values to be in for each of these molecules that we found. And so any of these values that fall out of those desired ranges are marked as outliers. So you can see there's a couple samples where the retention time of glucose was a little earlier than we expected. Like Nick showed with the email alerts, we can set up email alerting for these as well. For when an outlier is detected, sends out an email that says, this sample had a value that was outside our expected range. You might want to go look at it and see what happened. It could be that maybe your retention times are shifting as your column's degrading, or maybe the algorithm misidentified a peak for whatever reason. But this will just show you that, hey, there's an issue with this sample. You need to take a little deeper look. And this is a good way to get from where you're doing 100% inspection of all of your samples to where you're only reviewing by exception. And so only these outliers are the ones where you need the human in the loop, the scientist, to go in and kind of inspect what happened there, if it's something with the process or the sample preparation or something with the algorithm itself. So you can just find what samples these are and then go back to the chromatogram view and then see what happened there. Similarly, we also have a batch-to-batch summary page. So again, you can select your experiment and your molecule from this and pull up a couple more control charts. So here we have the R-squared and the slope of when we applied the calibration curve for each of these batches. And again, the same thing for the experiment summary page. We have expected ranges for each of these values set up. And if any fall outside of that range, again, we can flag it as an outlier. You can see here, we've had a couple outliers that were just outside of where we expected that slope to be when we applied the calibration curve. And so again, we can send out an email notification so you can inspect what happened in that experiment, what went wrong, or maybe if the algorithm needs to be tweaked a little bit further to fit your data accurately. And so that's kind of the chromatogram modular analysis app as a whole. And so I'll go ahead and pass it back to Nathan.
All right. Thank you, Olivia. All right. Well, thank you both for presenting that. Again, we're really excited about modular analysis and some of the potential it has. I'll highlight again everything you saw there. A lot of the power that comes from modular analysis is that that is all fully customizable as much as you need, but also linked directly to your instruments or the context from your e-liner limbs. So when we're talking about associating data with batches or experiments or samples, that is something that is fully taken from your existing data systems. It's not local to the instrument. We can compare data across every run that you've ever done, tracked in your e-liner limbs, or we can compare data between different instruments. We're not isolated to one instrument or context. So that's very powerful for giving more context, more insights. And then as you saw, we can predict as much as we can about these analyses as possible. So let's wrap up here and move to Q&A shortly. As a brief overview of how we think about the business case and some of the impact that this can have, I like talking about our case study with one of our clients, Solugen. Solugen uses an enzyme biology approach to produce biofuels and other petrochemical products in a fully green way. And for them, we integrated a bunch of different things, analyzers, an HPLC system that they had, bioreactors. And through the Ganymede platform, we automated the analysis. We cleaned up all the data, and then we were also able to automate the data entry of the final results into benchling, as well as automate all data movement from the raw data to those analyzed results into Databricks for them to do their own further downstream modeling. And this was very, very powerful for them because that automation both added a lot of scientific insight, but also saved them so much time. The amount of time that humans spend doing this manual work and repetitive analysis, especially when you're doing a pretty high scale assay, is tremendous. So we were able to 10x their sample throughput overall, mostly through removing those human components and save thousands of hours of scientist time that would have had to be spent otherwise. Want to also note on the domains that modular analysis is rolling out for, we're excited to work with everyone on all their domains. So if you have an assay that's painful, or you have an analysis that feels repetitive or could be improved or is a bottleneck for you on the scientific insight side, let us know. We love adding use cases, and there's this is all adaptable to really any analysis scenario. But the out-of-the-box ones that we're starting with are, you saw today, bioprocessing, chromatography, which we'll also dive deeper into in a future webinar. We also have a curve-fitting dose-response curve type of modular analysis tool, PCR, and our Aura capabilities, asset utilization, that we've shown in the past, are also now being adopted into modular analysis so that we can continue building a deeper and more interactive view of instrument utilization. Next up also, we have mass spec coming, as well as flow cytometry. So look out for more there. But with that, I'll turn it over to Q&A. I know there's a couple great questions already. I'll turn to Dinesh's question on what types of bioreactors there are that this can work with. So we can connect with really anything that has any clean file or OPC output or APIs. Ganymede is agnostic to any one platform or instrument. And so we have a connector library of hundreds of different instruments that we've connected to over time. We can also build new ones if needed. But as long as it has data coming out on OPC or files, no matter what the format is, our job is to go connect that and standardize it and roll it up. So any bioreactor out there, we've probably already integrated with it anyway, but there's nothing stopping us from working with new ones either. And then on the second question, in terms of who this is for, it's really for people running the wet lab assays, whether that's a biotech company or in a diagnostics context or any sort of lab. That's where we want to be. So we will automate those workflows, go in and connect to the instruments, and then can deploy modular analysis. But our goal is to help the scientists. And so as long as they're scientists or technicians or people doing things with their hands, that's where we'll want to be. So a lot of that is biotech, but again, it could be anyone with a wet lab. And let me actually pause this and check if we have any final questions on our end. Question from Demet: is there an easy way to scale the modular analysis product as our lab's needs grows? What sort of support and training do we provide? So yeah, when clients want to take on building these applications themselves, we love that. That's our ultimate vision, is to create a platform that people can use and adopt themselves. And so we usually will work with clients when we deploy to see if there's a super user we can designate that will be able to learn the lab's code system, and we can train them. So we have a whole developer training process that we'll go through that includes meetings and collateral. We have a whole docs website, docs.ganymede.bio, that's very built out for this. And so that's what we'll try to do, is incubate. Once we have some example integrations built, we'll try to incubate a person and some use cases that we can use to scale up our relationship and client capabilities for coding on this, and then work with them until they can be independent, and then hopefully set them off to their races on building more themselves. And we have one more question on LinkedIn, actually. What sorts of bioprocess modalities we support? I guess actually, Andrew, I might turn this over to you if you want to comment on like upstream versus downstream, or we've also talked about mammalian versus bacterial, things like that. What sort of modalities we're focused on now, and then what you would envision as being new modalities coming in the future?
Yeah, so our current bioprocess app is focused on upstream processing, and it's agnostic to the type of thing that you're culturing. So bacterial, mammalian, it's agnostic to both of those. In the future, we will have a downstream processing application that will look and feel very similar to the upstream bioprocessing application, although obviously for downstream processing. So to view purification, filtration data, formulation data, those types of things. The bioprocess application in its current state was really designed after the monoclonal antibody production use case, which is something that we see time and time again from a lot of our customers. So it seemed to be a good starting place for designing the app.
Awesome. And then we have a question from Chris on the chromatogram modular analysis. Many data sets on Chris's side have thousands of samples. How many samples can the visualization part of the process handle? Maybe the question for Olivia.
Yeah, so yeah, we can definitely visualize like on the thousands of samples. But as you grow and as you get more and more samples, we can set it up to where, it's only pulling in the data that you want to visualize. That's why we have like the experiment dropdowns. So we can modify that to whatever fits your process, so that we can pull in exactly just what data you want, and so it doesn't slow it down unnecessarily.
Awesome. And then we have a question from Chris on the chromatogram modular analysis. Many data sets on Chris's side have thousands of samples. How many samples can the visualization part of the process handle? Maybe the question for Olivia.
Yeah, so our current bioprocess app is focused on upstream processing, and it's agnostic to the type of thing that you're culturing. So bacterial, mammalian, it's agnostic to both of those. In the future, we will have a downstream processing application that will look and feel very similar to the upstream bioprocessing application, although obviously for downstream processing.
Cool. Well, that is all the questions. Oh, we have one more. When did the dynamic changes in modular analysis get written to Ganymede data tables? Are there separate data tables that need to merge later? Maybe also Olivia.
Yeah, so right now there's the publish data button in the modular analysis application once you're done with your modifications. And so that's when it would be written back to a Ganymede table. Right now they're written to separate tables, what the automated analysis finds and then what the modifications are. That can be modified to whatever makes sense for your process. If you want them written to the same table and then just having a flag of which ones were modified and by who. And so, yeah, you don't really need to go and merge them later. That's something that we can handle on our side of just writing them in the same format and either to separate tables if you want those separated or to the same one, just with kind of flags on them.
Yeah, and that's a great example of the customizability. We are hoping both that these out-of-the-box applications can be customized from there to suit your workflow, but also that they can be kind of a bombs up. Like you could use this to build a fully raw app if you just need some small widget to help with some sort of like an automation use case or reviewing a chart on something. So lots of flexibility. All right, well, that's all the questions for today. Thank you all for attending. We'll share over the recording as well. But as always, please reach out to us if you're interested in this, if you'd like to learn more or have other questions. We love to chat about it. We wouldn't be here if we didn't like talking about this forever. So thank you, Olivia. Thank you, Nick. Thank you, Andrew. And thank you all for attending. Have a great week.